What if the secret to stopping brain degeneration has been hiding in our cell's own cleanup crew all along? Groundbreaking research has just shattered conventional thinking about how we might fight devastating brain diseases like Alzheimer's and Parkinson's. Scientists have discovered that by supercharging a single protein called PI31, they can literally reverse neurological damage in laboratory animals. And here's the kicker: some mice lived nearly 4 times longer than expected. This remarkable breakthrough, detailed in the prestigious Proceedings of the National Academy of Sciences, centers around what researchers are calling the brain's molecular janitor, a cellular waste management system that could hold the key to preserving our minds as we age. But here's where it gets controversial. This discovery challenges everything we thought we knew about treating neurodegenerative diseases. To understand why this matters so much, let's break down what's actually happening in our brains when diseases like Alzheimer's strike. Think of your neurons as busy factories that constantly produce various proteins to keep your brain functioning. Over time, some of these proteins become damaged or simply outlive their usefulness, like old machinery cluttering up a workspace. In healthy brains, specialized cellular cleanup crews, called proteasomes, sweep through and dispose of this protein garbage. The PI31 protein acts like the foreman of this cleanup operation. It's responsible for assembling these proteasome crews and making sure they reach the far corners of your neurons, particularly the synapses, those crucial communication junctions between brain cells. When PI31 isn't doing its job properly, protein waste starts piling up, synapses begin to malfunction, and those infamous protein plaques associated with brain diseases start forming. A number of diseases, Alzheimer's, Parkinson's, are in fact diseases of synaptic dysfunction, explains Dr. Hermann Steller, a professor at the Rockefeller University who led this research. Now that we've shown how to eliminate unwanted proteins at the synapse, we hope this will lead to a revolution in treating common age-related disorders. And this is the part most people miss. The research team had previously discovered that patients suffering from various neurodegenerative conditions, including Alzheimer's, Parkinson's, and ALS, often carry mutations in the gene responsible for producing PI31 protein. This wasn't just a coincidence. It was a smoking gun pointing to a fundamental mechanism of brain degeneration. The current study took this revelation and ran with it, testing whether boosting PI31 levels could actually reverse the damage. Working with fruit flies and mice that carried FBXO7 gene mutations, known culprits in early-onset neurodegeneration and Parkinson's disease, the researchers cranked up PI31 production and watched something extraordinary happen. In fruit flies, severe movement problems caused by the genetic mutations simply vanished. The proteasome cleanup crews started moving through neurons again, clearing out the accumulated cellular debris. But the real shocker came with the mouse experiments. Even with just a moderate increase in PI31 levels, the mice showed dramatic improvements. Their neurons stopped degenerating, their motor functions returned to normal, and those characteristic tau protein tangles that define Parkinson's and Alzheimer's began disappearing. Most remarkably, the treated mice lived significantly longer, some nearly quadrupling their expected lifespan. The degree to which we can rescue the various defects in mice is remarkable, Steller noted, and honestly, that might be the understatement of the year. Now here's where things get really interesting and potentially controversial. This approach doesn't just target rare genetic disorders caused by FBXO7 or PI31 deficiencies. The implications stretch far beyond these specific conditions, potentially offering hope for the millions of people facing common age-related brain diseases. But let's pump the brakes for a moment. While these results are undeniably exciting, we're still talking about laboratory animals, not humans. The leap from mouse to human is notoriously treacherous in medical research, littered with promising treatments that never made it to the clinic. The research team isn't stopping here, though. Their next major investigation will examine whether targeting PI31 can preserve cognitive function in aging mice, a crucial step toward developing treatments that could maintain our mental sharpness as we grow older. We're extremely excited that this is relevant beyond our fly and mouse models, Steller concluded. The science implies that our findings may potentially, down the road, allow us to slow down cognitive decline as we age. This raises some fascinating questions that deserve serious discussion. If we can enhance our cellular cleanup systems to prevent brain degeneration, should we? At what point does treating age-related decline cross the line into human enhancement? And perhaps most provocatively, if this treatment eventually becomes available, who gets access to it? The discovery also challenges our current treatment paradigms. Instead of targeting the protein plaques and tangles after they've already formed, the approach most current Alzheimer's drugs attempt, this strategy focuses on preventing the cellular garbage from accumulating in the first place. It's like fixing a city sanitation system rather than just cleaning up after the trash has already overflowed into the streets. What do you think about this approach to treating brain diseases? Does targeting our cellular cleanup mechanism sound more promising than current treatments that focus on removing plaques after they've formed? And here's the big question. If a treatment like this becomes available, do you believe it should be considered a medical necessity or an optional enhancement? Share your thoughts. This is exactly the kind of breakthrough that deserves robust public discussion about where we're heading with brain health and longevity research.